Leaf structure and transpiration. First thing, in the structure of the leaf, you have the cuticle. The outermost layer of both the upper and lower surfaces is up here in the top. It's clear and waxy. It prevents water loss. So when you touch a leaf, it's that weird feeling, that harshness, or that kind of water-resistant touch of the leaf. Okay. Next, you have the epidermis. So then you have upper epidermis here. The layer of cells, one cell thick, provides protection for inner tissue. This is the inner tissue. Tissue is a word you use for a cluster of cells. These cells, epidermis, are clear to allow light to reach the photosynthetic tissue, which are the chloroplasts or the pigments in the plant. Finally, you have the mesophyll. Between the epidermal layer, it contains the palisade cells that are tall, tightly packed, that they filled with chloroplasts for photosynthesis. A palisade is another word you use for a fence in a castle. This is a palisade at the medieval castle. This is a Hyundai palisade. Yeah, go figure. So a lot of words that you see here in this plant are also words that you'll see in other uses. So your epidermis is like the skin cells in your body. It's called epidermis. Mesophyll, meso means middle, so there'll be mesophyll in the middle. Then you have palisade layer here. So let's give you a moment to copy these things down. So you can stop the video right now while you copy this down. I'll give you 3 minutes to copy this down. Continuing on with the video, the structure of the leaf, you have your stomata. This is the opening of the surface of the leaf for gas exchange. We'll go into that in more detail. They're basically little holes in the plant that allow the plant to accept gas. There's water vapor that passes out through these leaves also. But what happens is it basically is like the mouth of the plant. They used to breathe, inhale, exhale. There's special mechanisms to keep the stomata closed when the plant wants to retain water and retain gas, keep the gas inside of itself. But it also has mechanisms to open up the holes so it can get gas out or get water out. And we'll talk about that later. But within the plant cells, you always have a stomata or a little hole. Next, you have veins. These contain the vascular tissue. Vascular is a word you use for veins or anything that transports fluids. It applies to human beings. Like you have vascular veins in your body and arteries. Same thing in the plant. The xylem carries water and minerals upward. I think of XYZ, xylem, because it's XY, right? So you think of WXYZ and W being water. The xylem is for the water. The phloem carries dissolved food throughout the plant. Phloem is like sugar. So think of like flow. It allows thicker things to flow through the plant. Thicker things like watery sugar or watery minerals, potassium, nitrogen, etc. So when you look at the structure of the plant, you have the cuticle on the outside the plant, which is a waxy surface protecting it. Below that, you have epidermis. You have a lower epidermis because it's the top and bottom of the plant. Then you have your xylem and phloem inside the plant. Then you have your stomata, these little pores that let air in and out or water in and out. We'll talk about the guard cell later. So in your drawing, you should just have the basic things right here, like the bundle sheet cell around the phloem and xylem, the upper epidermis, the mesophyll, the palisade layer, which is like the fencing of the plant, and also referred to palisades of a castle. You have a spongy layer below that, and so that helps fill in the plant and create support. So I'll give you some time to copy it down, no pressure. 10, 9, 8. I will just stop the video and let you copy it down right now. Alright, moving on. Transpiration is the loss of water from a plant by evaporation. So transpiration is a fancy word for evaporation. Evaporation is when a liquid goes to a gas. So within a plant, the liquid in the plant goes to gas and evaporates out of the plant. Water can only evaporate from the plant if the water potential is lower in the air surrounding the plant. What does that mean? You have less water in the air around the plant. So the plant will only lose water if the water in the air around it is less. This is diffusion. You're going from a high concentration to a low concentration, similar to what we talked about before. They're using this word water potential, and it sounds really scary, but all it means is the water content of the air. So basically, if there's more water in the air, the plant will not lose water because there's more water in the air. The water in the air wants to go into the plant. Remember, in nature, it's all about balance. When you have less, nature tries to fill the gap. If you have high concentration of cologne in one area, nature wants the cologne to spread out and even out. Same thing with water in a plant. Most transpiration occurs by the leaves, and it's by the stomata, those tiny little pores in the plant, which we'll talk about tomorrow or the day after. How transpiration is measured? Good question. This is a potometer. No, it's not a potometer. It's a potometer. Anyway, you take the plant, you hook them up to a potometer, which measures the amount of liquid in the plant. The leafy shoot is cut underwater. That prevents gas from getting into the plant, because like flowers you give to your girlfriend, you want to make sure you cut it and then put the flower in the plant in the water, and you want to make sure it's a fresh cut. That way there's no air blocking the movement of water in the plant. You have plastic tubing, and then you have a capillary tube. What's a capillary tube? A capillary tube is basically a very small tube that moves a very small amount of water at one time. Water is sticky. It likes to stick to each other. It's called adhesion. We talked about properties of water in the first semester. So when one water molecule moves along the capillary tube, it tends to pull water along with it. This is passive transport. It does not require any ATP. So that's your capillary tube. Airtight seal, because if you have air entering the plant, you're going to lose water to the air. That's called evaporation. We want the transpiration of the plant to measure how much liquid in the plant is actually moving. Make sure your air seals are tight. That's why we're doing the virtual lab. In a normal lab, it's very hard to measure this because you have so much room for error. If you have a loose air seal, you're going to lose water to the air, and you're not going to get an accurate measurement of how much water is moving through the plant. You have a graduated scale, so you can measure the amount of water. And the movement of meniscus is measured over time. Remember, the meniscus is that little part of the liquid here, like the little smiley droop of the liquid. So water evaporates in the plant, and as it evaporates, it moves the water up the tube, and you lose water, and that's what you measure. The volume of water taken up at a given time. The other problem with doing this in a real lab is probably needed it to be like 1 hour, and we don't have the time for this. That's why we're doing a virtual lab after this. The rate of water loss from the shoot can be measured under different environmental conditions. Limitations. You measure water uptake. The cutting the plant shoot may damage the plant. If you damage the plant, it affects the ability of the plant to transport water. They have no roots, so no water resistance to water being pulled up. So, in the lab, we'll pause this. We'll see if everyone else is ready. But if everyone else is not ready, you can talk to me a bit, and then you proceed to the virtual lab.